All right, well, hello everyone, and welcome to today's edition of the EV Journal Club. So today we're going to be talking about looking at extracellular vesicle uptake and the uptake of EV cargo. And so today's presenter is Franziska Haderk. So she's at UCSF right now, San Francisco. She previously was working in Heidelberg, Germany, where she did some work on extracellular vesicles. And so, Franziska, thanks so much for joining us today and being willing to present this paper. And I'd also like to introduce two of the authors of the paper. So the first author, Olivier de Jong, and then Peter Vader is also the corresponding author of the paper is on the call too. So would you just like to say a couple of words about this paper and all the work that went into it?
Yeah, of course. Thanks, Gavin. First, thanks very much for having us, and yeah, for taking the time to discuss our paper. It's a big honor. I think I don't want to steal all of your thunder, of course. So I think for explaining the amount of work that was put in, especially by Olivier, I think he can explain best himself.
Yeah, so first of all, once again, thank you very much for discussing our work here today. So yeah, we set out to find a way to be able to measure functional RNA transfer, and fortunately we were successful, but yeah, it took us quite a few years, and well, we're happy with the see how you guys received the paper.
Well, we're thank you very much, and we're excited to now look at what was in this paper now that it's out. So, Franziska, thank you again, and I'll hand it over to you. And I would just ask that everybody please put your comments in the chat box, any questions that you have, and at the end of the presentation, we can unmute you and have a nice discussion here with Franziska and the authors. Franziska, thank you.
Yeah, thank you so much. And again, thank you for putting this journal club together. It's a great opportunity. It has been so much fun over the last month, and I hope many more months and years to come. I think as Kenneth already said, I'm a postdoc now at UCSF, studying drug resistance to targeted inhibitors, and have done work on exosomes and their role in the tumor microenvironment crosstalk before. And at the end, hope to bring it together and understand how drug resistance can also influence the microenvironment in my future work. Yeah, I'm presenting this really cool paper today, which was published in Nature Communications this year by Olivier and Peter, with also senior ownership by Matthew Wood, which describes this beautiful CRISPR-Cas9 reporter system that is allowing us to study vesicle-mediated RNA uptake and processing. And I also just have to put in this thing from the recent Nature Outlook that shows extracellular vesicles within our body and how they are these like super vehicles of RNA that can mediate cancer or in general cell crosstalk within the body, even at a distance, and how I see also exosomes and extracellular vesicles in our bodies being this really cool shuttle molecules for RNA. So that's why I think it's so important to develop reporter systems that allow us to understand the whole mechanism behind it in more detail. So as I said, yeah, we have a continuous need to understand vesicle-mediated RNA uptake and processing. There are some also very cool reporter systems out there already. For example, the QLOX-P system, I think everyone is aware of. However, they have been based more on mRNA-mediated transfer. So in this case, we have a donor cell that is expressing the pre-mRNA, and when the pre-mRNA is packed into extracellular vesicles and taken by reporter cells change their fluorescence from basically red to green. And I've just put in some of the in vitro work of this paper in case you want to discuss this later. And you can see it in particular in the upper panels how in an in vitro co-culture system, we have approximately 1 to 10% reporter selectivity, and we can actively show that the mRNA, pre-mRNA is taken up in reporter cells and induces green fluorescence. However, the system is limited by studying large RNA, mRNA transfer, and of course also biased by the contribution of the mRNA and the Q protein contributing to reporter selectivity. Plus, we all know that vesicles are full of small RNAs, and it is really important to study a small RNA transfer of small structural RNAs independent of bias of RNA transcription and translation. So the authors have designed this really cool CRISPR-Cas9 system. I think everyone is aware of CRISPR technology by now. I just have put in a very short paper to explain it again. So what you basically do is you present the Cas9 or deliver the Cas9 into your target cells together with guide RNAs, and then direct the Cas9 to your DNA sequence of interest. And the Cas9 molecule as a nuclease or protein as a nuclease then cuts the DNA and induces a DNA double-strand break. This can in the cell either be repaired by non-homologous end joining, NHEJ, or homologous directed repair in presence of a template. So in presence, for example, of your sister chromatide. And for our system in particular, since we don't have a template present, the non-homologous end joining is the repair mechanism that is predominating here. Yeah, and now jumping into the supply system, the core system, it's a very interesting and very smartly designed what a construct presented here in A combines is the expression of mCherry, followed by a Cas9 targeted linker. So Cas9 is encoded by the linker. And since we're talking about, and then after this follows the sequence of EGFP open readings, either one or two nucleotides out of frame. So basically what happens is if you have a guide RNA that targets Cas9 to the linker sequence and you get a cut that is repaired by non-homologous end joining, in 80% of the cases you have a frame shift of one or two nucleotides that then allows the EGFP to be expressed. So in any reporter cell that basically shows guide RNA activity on their stoplight reporter system, you get green mCherry and GFP fluorescence co-occur within the reporter cells. And first of all, we validated this stoplight system in the HEC293P reporter cells by either co-transfecting non-targeted guide RNA or targeted guide RNA against the linker, and showing that you have a strong induction of fluorescence when you co-transfect targeted guide RNA, as depicted in B by confocal microscopy as well as in C by flow cytometry. Now, how can you use the system to study vesicle-mediated RNA transfer and RNA processing? You simply basically split up the donor and the reporter cell. The donor cell that is providing or expressing the guide RNA, RNA has packaged into extracellular vesicles and is then taken up by a reporter cell that is expressing the Cas9 and the stoplight reporter. And when co-culturing basically the donor and the reporter cell, in this case, this is the breast cancer cell line MBA-MB231 donor expressing the guide with the HEC reporter cell expressing Cas9 and the stoplight transfer, you see that you can induce green fluorescence when you co-culture with donor cells that are expressing the targeted guide RNA. So there is targeted guide RNA that is packaged into vesicles and then transferred into the reporter cell and induces reporter activity. As you can see in figures E as well as in F. And what is also very interesting is that this is basically a dose dependency. So having different donor reporter cell ratios, you can get an increase of reporter cell activity. Further, the authors very clearly point out to the fluorescence that they can achieve. There's I think it's a very reliable system and well also showing the dose dependency about tunable system. However, the fluorescence reporter activity you can achieve is approximately 0.2% of reported cells that are positive. And we will discuss this in the future figures also, what are limiting factors that are resulting in 0.2% positivity here. What is really, really nice is that the authors have also shown the reproducibility of the system across a multitude of reporter as well as donor cells. So they have generated on top of the already presented results 5 more reporter cell lines. I only have presented 2 here, where they can also reliably show that the system is working, that you can induce reporter cell activity in a dose-dependent manner. Similarly, they have generated a multitude of donor cells, in this case 3 donor cells, and interestingly here, while 2 of the donor cells are able two of the donor cells induce reporter cell activity, there is one cell, the HEC2 and 3T donor cell, that is actually not inducing reporter cell activity in HEC2 and 3T reporter cells, which is very, I think, interesting to note that we, of course, have few limitations regarding which cells can take up the vesicles, what are the target cells of the vesicles. And I think this can, again, give us a huge data regarding the functionality and which cells can take up vesicles and which can't, or what is the crosstalk between different cell types. Further, authors did a lot of experiments to really show that the guide RNA transfer is predominantly performed by vesicles. The first step was to rule out cell-cell fusion. The authors here did this in a very, very cool way by co-expressing luciferase in the donor cells. We have donor cells that are expressing guide RNA and luciferase, and that you co-culture with your reporter cells in total for 7 days. And then you sort by flow cytometry, by separating the different populations of your donor cell and the reporter cells, either positive or negative for each GSP. And you then, about cell fusion, you would likely become reporter cells that are not only positive for EGFP, but also show luciferase activity. And I think this is done really nicely. So the authors have shown that the donor cells show an increase of luciferase positivity with an increase of cell number. However, comparing the luciferase activity in donor cells to reporter cells, either EGFP positive or negative, there's basically no activity in the reporter cells. There's no luciferase transfer, and there's likely no cell-cell fusion. In addition, they have also shown that the luciferase activity is present in the cell lysate, but not in the extracellular vesicles. There are hardly any luciferase activity in the vesicles, but you can see it in cell lysate and also the isolated protein fractions. Further, we have now mostly talked about direct co-culture assays. The authors also showed basically that this process happens in a cell contact independent manner by using like transwell assays, having reporter cells in the lower transwell and donor cells in the upper transwell, with basically a transwell insert that allows the extracellular vesicles to transfer into the lower well and then being taken up by reporter cells. And again, when you compare untreated reporter cells to reporter cells that are in the transwell co-culture, either with donor cells expressing the non-target guide or the targeted guide, you see that there is a specific activity in the co-culture with donor cells that are expressing the targeted guide RNA. And this can be inhibited by the GW4X9 inhibitor. So when you inhibit vesicle generation, you can basically inhibit the guide RNA transfer either in the transwell or in the co-culture assay. Further, one step ahead, the authors then went one step deeper and showed that the guide RNA transfer is mediated by extracellular vesicles by directly using extracellular vesicles to treat their donor cells. They have isolated these vesicles from the guide RNA expressing donor cells by size exclusion chromatography, and then characterized vesicles by Meiss's guidelines, including NTA profiling, showing a mean size of 80 nanomolar, or a peak size of 80 nanomolar for vesicles, showing positivity for vesicle markers such as CD9, CD63, and being enriched in the vesicle fraction compared to cells, but absence of markers like calnic, and I don't see the other H2B, histone 2B, being absent in the vesicles compared to cells. We see this electron microscopy picture corresponding with the NTA profiling. So we have a lot of vesicles that are in a range of 100 nanomolar and some bigger ones. And we have the RNase protection assay that shows that if you treat your vesicles with RNase or proteinase, the guide RNA is still protected. However, if you disrupt vesicles with detergent, then the RNase can basically access the guide RNA and the RNA is degraded. So I think a very well characterized isolation of vesicles. Further, the authors also asked the question, how many guide RNAs are actually present within the vesicles? And this they did by basically having a standard curve of synthetic guide RNA and comparing this to the presence of guide RNA in the vesicles, showing that we approximately have one guide RNA for each basically 500,000 vesicles, which I think is a very interesting fact to know that we have one guide RNA per 500,000 and tested whether vesicles can be taken up in reporter cells. On the left, you first do basically the control, where you have PKH67 labeled vesicles and their uptake in HEC293T cells. We see uptake of vesicles in HEC293Ts. And then on the right, you see the uptake in the reporter. So this basically use vesicles either isolated from donor cells that are expressing a non-target guide RNA or donor cells that are expressing a targeted guide RNA. And we see the induction of reporter cell activity upon treatment with vesicles having the RNA. And again, this is a dose-dependent mechanism. So if you apply more vesicles, you get more reporter activity. And I think what also interesting is if you in the same experiment use your isolated vesicles as well as the isolated proteins, you see the reporter activity being transferred on vesicle treatment, but not up on treatment with the isolated protein. Again, showing how selective the system is to really show the RNA transfer by extracellular vesicles. And yeah, so before we jump into this slide, maybe to sum up, so we have the stoplight reporter system that can be well used to monitor extracellular vesicle RNA uptake and transfer either in co-culture assays, in cell-free assays, or upon treatment with vesicles directly. And we also have very nicely ruled out that there are events like cell-cell fusion or any protein-mediated guide RNA transfer that contributes to reporter cell activity, showing really how well this system can be used to study guide RNA or in general small RNA transfer by vesicles under physiological conditions. And now with this figure, the authors really asked the question, okay, can we understand more about the mechanism of RNA processing and vesicle processing in the target cells? They did this by performing siRNA-mediated knockdowns, either in the donor cells, as you see in the upper panel, or in the reporter cells in the lower panel. So you basically perform a knockdown of your candidates of interest in either cell, and then perform co-culture assays with the respective others, so either with reporter or donor cells, and analyze how much reporter activity is achieved. I think what is almost a proof of concept is if you knock down proteins that are involved in exosome biogenesis and exosome processing within the donor cells, you see reduced reporter activity in the reporter cells. So less exosomes are generated, less reporter activity is observed. And then what is, I think, in my opinion, really novel and really cool is that you can also use it the other way around. You knock down candidates in the reporter cell and see, okay, what is actually inhibiting the exosome-mediated RNA. And there are all of these four molecules I pointed out here, caveolin-1, RhoA, PEG1, and REP1, either RhoGDPases or RhoGDPase effectors. So I think that is really also a tool to understand in the reporter cells what is important for vesicle uptake and for uptake of the RNAs and processing of vesicle cargo. And the authors, again, this was in co-culture assays. The authors then again went one step ahead and checked whether the same phenotype is observed when you treat reporter cells with vesicles derived from donor cells that went derived from donor cells. So they compared reporter cells that are either have a non-target siRNA knockdown or caveolin-1 siRNA knockdown, and how much reporter activity can be achieved. So you have reporter cells, you treat them with vesicles. This has been done over 6 times. And then you give them another 2 days before you measure reporter cell activity. And again, you can see caveolin-1 knockdown in your reporter cells significantly reduces the vesicle-mediated reporter activity. And again, this can be expanded to other important candidates, which I hope we can also discuss today, including IgG1, other important proteins that are involved in intercellular membrane trafficking, like ROC5 and ROC7, and ROGGPases in particular, also ROC. And one step ahead, the authors also used the ROC inhibitor Y27632 to show again that this is a tunable, and if you inhibit ROC, you again get reduced reporter cell activity compared to vehicle. And last but not least, the experiments you have seen so far have all been conducted under guide RNA expression under polymerase III promoter, U6. The systems are usually more tunable and, for example, also involved in doxycycline inducible systems. So I think the authors really have done a great effort to change the system into a polymerase II mediated expression system using also self-cleaving ribozymes to inhibit secondary modifications of the guide RNA that again could inhibit function of the guide RNA. And they have compared the standard polymerase III system to either non-inducible, so standard expression polymerase II system, EF1, or a doxycycline inducible TET-on system that is kind of set up, and you can see the results on the right. So comparing either the polymerase III U6 or polymerase II EF1 alpha reporter or like guide RNA expression system, you see the same reporter activity, and again, it's inducible comparing on the far right minus and plus dox treatment. So just to summarize, I think it is a really cool system and a really reproducible system shown across a multitude of reporter and donor cells and multiple systems to study vesicle-mediated guide RNA transfer. There is no background from cell-cell fusions or protein-mediated transfer. It's really specific for vesicle-mediated transfer. The packaging of guide RNA is to a certain degree limited. There's one guide RNA in every 500,000 vesicles, but this also allows monitoring of biologic processes involved in vesicle processing at a physiologic level. So I think the authors, and maybe you guys can comment on this, you really actively decided against overexpressing or changing packaging and forcing guide RNA into vesicles to be able to study this all under physiological conditions. And we have seen a lot of experiments that this is tunable, it's dose dependent, you can inhibit it. So I think it's a very reliable system to study physiological processes. Further, they also have shown very novel regulators of vesicle-mediated endocytosis and processing in the reporter cells. And we have seen that the functionality of the system is also maintained when the polymerase II promoter is used for the guide RNA expression. And with this, I hope for a good discussion and also contribution from Olivier and Peter. Discussion points I have put down, which I think are interesting to discuss, is the limited, and not in a negative way, limited positivity of reporter cells. We have 0.2%. I wonder if this is just like, okay, this is an average how small RNAs are kind of like transferred into the exosomes, or is there something that hampers packaging, or also is there some limited accessibility of the stop-like transporter system? Again, I think this is showing physiological levels, and also comparing it to other systems like Prelox-P, they are only a little bit above this positivity. And yeah, I think what this system also offers and what we might discuss is like, what are differences in the reporter cells? Is this uptake stochastic, or are reporter cells also showing differences that allow some to take up more vesicles and some to take up less vesicles? Yeah. And so I think that's it from my side, and I hope for discussion.
Great. Well, thanks so much, Franziska. We have had several questions that have come in the comment box, but maybe we can just start with these discussion questions that you have proposed, and maybe you and the authors could discuss these first.
Yeah, I'm happy to start answering or discussing the first question. Well, first of all, Franziska, thanks very much for explaining our work so carefully. I think you did a great job there.
Yeah, thank you for the great work.
So, I mean, about this physiological loading, we think that the numbers that we see mimic endogenous RNA levels. in vesicles if you look at specific RNA species. So that's why we try to stick to this and not do any tricks for loading, in which we would manipulate and maybe make the system more efficient, but make it more difficult for ourselves to study the biology and physiology of all this. So, so what hampers packaging, basically this is a non-natural guide RNA. So we think that the loading is spontaneous and random. But of course we have no proof of that, but that's how we envision that this is working. So what is then the limiting factor in terms of positivity in the receptor cells? And we think it directly has to do with the loading efficiency. So what we're currently doing with this system, and that's actually why we, amongst other reasons, got interested in this vesicle field in the first place, is that our lab has a strong focus on RNA therapeutics and RNA delivery. And what we can now do with this system, and actually Dan Murphy will present a post on this at the online ISAF meeting, is compare the efficiency of extracellular vesicle-mediated RNA transfer as compared to synthetic nanoparticles. So lipid nanoparticles that are currently available, being used in the clinic, but also satisfaction reagents, RNA-MX, etc. And when you do that comparison side by side, we actually see that these vesicles do deliver that small amount of RNA that they have in a highly efficient manner. So we think that although of course there are still limits to the RNA delivery efficiency in terms of endosomal escape, perhaps if you compare it to other systems out there, they are doing this in a highly efficient manner. So we think the biggest hurdle for this positivity, let's say, for the amount, the percentage of green cells that we observe, is in fact this loading efficiency.
So how do we improve that without losing function?
Yeah. So the question is, I mean, how do we improve that? That depends on where do you want to use the system for. So there's at least for if we talk about what we're pursuing is in one way see if we can make this system even more physiologically relevant by trying to do this in 3D or try to see if we can use this system in vivo. And for that, we actually don't want to engineer the system too much anymore. I think the sensitivity on the reporter end is reaching its limits. I think it's a really, really sensitive system. I don't think there's much to be gained there. Of course, you can improve the loading. And from a drug delivery perspective or from an engineering perspective, that is of course highly relevant. And there's several ways in which you can envision doing that. And there are several labs in the world that are trying this as well. But for the biology and in the investigation of RNA transfer in a biological manner, I don't know whether we should do that really.
Yeah, I think that's also an interesting point because from my prior work, you can see that also guide RNA or like RNAs in part are also recognized by toll-like receptors when they're integrated by vesicles. It doesn't have to be true in all cells, but in particular in antigen-presenting cells like myeloid cells, that could of course be triggered. And I think if we overload the system with RNAs, we often maybe lose the balance that the physiological system has in place to be so efficient, right? Like we have maybe side effects of inflammation that we can't even foresee.
Yeah, for sure. I mean, I'm not claiming this is physiological, of course. I mean, it's a single guide RNA that is randomly carried into a vesicle at some point, and it represents of all RNA molecules. Then again, all information that is out there, I think now suggests that vesicles, when they're taken up by the recipient cell, follow the endocipient route and need a fusion event to unload their cargo in the recipient cell. And so does all our RNA molecules. So in that respect, I think it serves as an okay model. So I'm also looking at the comments and I'm trying to answer some of those at the same time. But yeah, you're right that if you can overload and yeah, maybe lose efficiency, I think. Although we don't have evidence for that really.
Yeah.
So let me just interject one question, because we've had some papers that we've gone over in EV Journal Club about microRNA transfer, and we even had a little bit of a debate a few weeks ago about it, how much of that actually happens. So now with this system, you have a very sensitive system, and you're still kind of pushing up against the limits of that sensitivity. Do you think that there is a way, a good way out there, or something that you can envision where we could better assess the uptake of microRNAs?
Yeah, I've had this discussion a couple of times already. I think that it's very difficult to adapt this system for microRNA transfer. We have been thinking about very complicated systems, but no, at the moment we don't have a good answer for that. Olivier, do you have any more ideas on this?
Well, I mean, I wasn't there for the discussion that you're within the journal club, but I mean, one of the main limiting factors, of course, is that you have to study the effect of the microRNA on bulk cell populations. I think the strength of this system is that you can really see activation in one cell at a time. And I think unless you come up with a trick to be able to do that with functional microRNA transfer, it will be really difficult to see any type of effect, because you always have variation of expression levels in different cells within the same well. So yeah, that would be a very challenging thing to do.
I would hope that this system, for example, would allow us to study pathways and biology, and hopefully after, if we see targets or pathways that are involved in vesicle-mediated RNA transfer within our system, that we can use that in other systems where people study microRNAs, whether they can validate that in their own system, for example, and whether the same findings hold true for microRNAs. And that's currently very, very difficult to do.
Great, thank you. So Franziska, what's next? We're at the uptake?
Yeah, I think that was for me a question when I saw that, like, okay, you have always this little clusters of reporter cells and confocal images that take up vesicles, and then you have some cells that don't take up vesicles. And I think the system would also allow to kind of like do some phenotyping regarding what makes the reporter cell that takes up a vesicle, or like a guide RNA vesicle, different from the ones that are maybe less likely to take them up, or is this just stochastics, right? Like, is it pure stochastics, or is there some changes in phenotypes within the reporter cells that allow better uptake? And I think in some part that also comes with the siRNA experiments you are showing with the road GTPases, that if knocked down, reduce uptake.
Yeah, so it is in that sense, it's unfortunate that the loading efficiency is so low. So we can't really study specifically which cells have taken up the guide RNA, for example, and whether these cells behave differently, because the cell next to that one may have internalized the same amount of vesicles or the same types of vesicles. It just happened that these vesicles by chance didn't have that particular guide RNA, and therefore didn't turn the greenhouse system. So to answer this question with this reported system, I think is really difficult to do it on a single cell level. We thought initially to indeed sort the green cells and then look at, well, which what genomic changes are there as compared to other cells. But because the efficiency of loading is so low, we can't really do that. If you look under the microscope and we label our vesicles fluorescently, it seems that all our cells are taking up the vesicles just fine. So it's not that the uptake is that specific among cells within a well, for example. We do see that some cells respond better to in a co-culture within in the crossfire system as compared to other target cells. But whether that's because the donor cell sheds more vesicles, or they package more guide RNAs, or the target cell internalizes more or processes these vesicles more efficiently, there's of course questions that we're trying to answer, but it's really ongoing work.
Mhm.
And I think, yes, I think that this question about the cells also leads us into another line of questions that have come up in the comment box. Maria Fernandez, for example, asked about the pore size in the experiments. Ganesh Shelka is asking about the whether apoptotic bodies might be involved here. Dolores Davizio is asking about EV heterogeneity, and then Anne Wayman also is asking about some pathways here. So I'll let these people unmute themselves if they'd like to expand on their questions. But I think that the question then of EV heterogeneity is really important here too. So you're looking at a general population of EVs. One of our first speakers in the EV Journal Club was Wyatt Vreeland from the U.S. National Institute on Standards and Technology, and he had done some experiments with liposome uptake, where he had he had made populations of liposomes that varied in size, and found that their uptake was dependent upon their size. I guess that's something good to discuss here: size and or other features of the EVs that may prompt better uptake. And again, Dolores and Ganesh and so forth, if you want to unmute yourselves, feel free to do that. Everyone can unmute now.
I mean, regarding the heterogeneity, of course, that is a highly interesting topic. Heterogeneity in size or biogenesis pathway. On 6 consecutive days, using quite a large starting volume from which we isolate our vesicles. So at this point in time, it's really not feasible for us to make subsets out of that EV pool and then perform these experiments at the moment. So in order to do that, we would first have to improve our loading efficiency quite drastically, I think. So in terms of size, I'm aware of the fact that liposomes or lipid nanoparticles in general show different uptake pathways with differing size. Whether that's true for vesicles, we don't know. And at the moment, it's just not practically possible to study that with this system. The same holds true for separating apoptotic bodies from different vesicles. If you talk about separating size, what we can do, of course, and that is actually something that we do in the paper as well, is that we use siRNAs to knock down proteins involved in various biogenesis pathways. We do, for example, show that Rab27A knockdown significantly decreases the RNA transfer. And if we believe that Rab27A is more or less specifically involved in the biogenesis of exosomes or other types of vesicles being shed from living cells, that would rule out to some extent that at least not all the signal that we see is coming from apoptotic bodies. I tried to answer 2 or 3 questions in once now, but perhaps someone else would like to ask an additional question now.
Can I, hi, this is Dolores. Very nice presentation and very nice paper. So I have a question for Peter. Did you say, so you haven't tried to isolate large and small EVs at the best of our abilities and see at least if the guided RNA expression is higher in one or the other?
No, we haven't done any of those studies. I mean, I guess I can predict it because the packaging is random. The chance of them being ending up in a larger vesicle would also be larger. But we haven't done any of those experiments in which we try to separate different subsets of EVs. And like I said, it's also a practical limitation at the moment, because we already have to really push the limits of the reporter system at the moment when we isolate the whole bunch of vesicles. So in order to do those types of experiments, we need to work on the loading a little bit. Yeah. Or find a combination of cells that communicates much more efficiently. That would also help.
Okay.
So I see Esther Nolta has a suggestion here. Esther, do you want to bring this up?
Yeah, sure. Maybe it's a little bit of an overlooked concept here, but I do understand that the most likely explanation of the data may be that the efficiency of loading of the guide RNA is quite low. But I think we should also not overlook the idea that maybe it's also the heterogeneity in the acceptor cell population that plays a role in the efficiency of delivery. And since you have such a nice system, it may not be too difficult to maybe even do a single cell sorting and do single cell omics on this to see whether maybe there is something special about these GFP positive cells. What do you think about this?
Yeah, thanks, Esther. Yeah, I mean, it's something we've been thinking about, but the problem I see with that approach is really that because the loading is so inefficient that, I mean, almost all other cells in the wells that we have, even the ones that don't have GFP, they will have taken up the same number of vesicles, potentially the same types of vesicles, and just by chance, because we have only one guide RNA per 400,000 or 500,000 EVs that bears this guide RNA, only by chance that some of these cells then turn green. That's at least what we expect because the loading efficiency is that poor, basically. So that's why I foresee difficulties getting those cells out of the noise from all the other cells.
Yeah, it might be, but it may be something simple like a cell cycle stage or whatever that plays a role here and that you may be able to figure out. But I do understand your arguments that it could also be difficult. So yeah.
Yeah, true. Yeah.
All right, let's see here. I think that Anne, is your question answered? I think you had a few other points here that you wanted to bring up. Anne Wayman.
Yeah, I was mostly just wondering if phagocytosis was really involved, because integrins are involved in phagocytosis as well as the whole RAC and Rho pathways. So that was sort of surprising to me to see phagocytosis involved in something so small, really. And then I was just also wondering if you looked at clathrin or any clathrin pathways like AP2.
Yeah, so related to the phagocytosis and the integrin and caveosome that we've seen implicated, I mean, we don't know whether at this stage whether these proteins are truly involved at the stage of uptake. Also the GGPases, for example, I mean, they could be important at the stage of uptake, but of course later on during trafficking, also a function for some of these proteins has been described. So at this point, we really don't know where in the cascade we're looking. This is something, of course, we're pursuing, but we also can't rule it out. So we don't know whether it's the large EVs that are causing the effect that we see, or whether they're small ones that we have no idea about. But this is something we're looking at the moment. And then, do we think there are multiple uptake pathways? We think there are multiple uptake pathways involved, for sure. This is also something we've shown in the past. Using fluorescently labeled vesicles, there we found no role for clathrin mediated anisotoses for a specific combination of cells. But we did find roles for caveolin-mediated anisotoses, other clathrin-independent pathways, as well as macropinocytosis. But that was using fluorescently labeled vesicles that we then added to these cells. This reporter system would allow us to take this one step further and to look into not only the uptake, but also the trafficking downstream. Now we have some of these hits, but we still need to find out then at which step of uptake, trafficking, anisotropal escape, cargo release, etc., they play a role.
Great.
What would be Yeah, I think one question is also like, do you plan to also go in vivo and see if despite like the loading or the physiological levels of guide RNA in the vesicles, do you think you can employ this in similar in vivo level models as with the QE-LOXP system?
Yeah, we really want to do that. We're working actually with Jong and Vader, who first described the QE-LOX system for vesicle-mediated transfer in the tumor models that he has. He observed indeed a bit more efficient transfer in vivo as compared to what he found in vitro. So we hope to see the same. It's something we're pursuing. It's not that straightforward because we have a difficulty with the Cas9 that seems to be repressed a little bit in our mouse models. But it's something we're looking into and we're really keen on pushing. Yeah. So if you have ideas for that also in your models, I'm happy to hear them.
Yeah, happy to connect. It's really cool.
Franziska, did you have any other models that you wanted to bring up?
I think this was it from my part. Like, I think the most exciting was really also the correlation to QE-LOXP, as we just discussed, and the benefits of the sgRNA, small RNA system compared to mRNA system and their potential use in vivo. And yeah, the questions have been great. So I think one question would be maybe the therapeutic options and how we can learn here for RNA therapeutics and extracellular vesicles. Someone wants to comment on that?
Yeah, so this is what we're trying next is first to establish how efficient vesicles really are. And this is something that actually was the research question that led to the discovery of the system. This is what we wanted to do in the first place, to see how vesicles deliver their RNA, how efficient, but also if we assume that it was efficient, because it seems to be what nature uses for RNA transfer. If it is efficient, how would they do it, and what can we learn from that? So as I mentioned, we have some data now that suggests that vesicles can actually deliver that guide RNA highly efficiently as compared to the state-of-the-art synthetic nanoparticles. But why they do that and how they do that, that is a major question. There's now some literature coming out that models endosome escape. Inge Suhorn had a nice paper coming out on this endosome escape. So I think there's now more and more information coming into the field that suggests indeed that these vesicles can do it in an efficient manner. But how they do it and what we can learn from the vesicles and use that directly to improve delivery, I think that's still a bit early days.
Great. Ganesh, I see you have a question.
Yes.
A couple.
Yeah, I have a basic question regarding designing EVs for therapeutic purposes. So one of the things that is interesting for me is that the orientation of the RNA, whether it's luminal or surface, if you're considering the type of delivery for the therapeutic function. So, and it also explains some of the mode of delivery inside a cell. So do you have any ideas on that, or just something? So orientation of the RNA.
In our system, the guide RNA is luminal. We show that by proteinase, RNase, proteinase, and triton treatments. Correct. Thanks. Yeah. So this figure E shows that in this case the guide RNA is luminal, and I think, but that is a bit speculative. I think there's evidence from previous papers from our lab, but also from other labs, that for efficient vesicle-mediated RNA transfer, the RNA has to be in the lumen. And it makes sense if you think about the fusion event, that if that is indeed the mechanism through which vesicles unload their cargo into the cytosol, then it has to be in the luminal site of vesicles. So we also have a couple of years ago published on cholesterol-tagged RNAs, for example, that we incubate with vesicles and then have them on the surface. We did find that vesicles can carry those RNAs into cells and somehow induce knockdown, but that was highly inefficient in contrast to what we see here, which is highly efficient. So I think from a therapeutic point of view, you want your RNA, your therapeutic RNA cargo in the vesicle lumen.
Yeah, and I also think that like the reporter activity, like there are some systems which have RNA reporters that can be on the outside of the vesicle, but this is usually the fluorescence label, like the RNA itself is fluorescent. And I think here it's really in your reporter system about the function that the guide RNA has to be delivered functionally and then change, like actively induce Cas9 cutting within the reporter cell to get the fluorescent signal. And it's really nice function analysis, like of RNA, efficient RNA uptake and functional downstream effects.
Thanks.
Patrick, you had a question about stability. Patrick James.
Hi, thanks, Ken. Thanks for the fantastic presentation. Yeah, so just wondering, there's an open question, what the stability would be based on the pathways you observed. You know, how much of the actual guide RNA do you think could that be affecting what you're seeing as well?
Yeah, that's a good question. We don't really know this, the stability of the guide RNA in the endosomes, or because we don't know where it really escapes and makes it into the in the cytosol. So this is very difficult to answer. I mean, if we do rough calculations, we think that we're adding maybe between 10 and 50 guide RNA molecules per cell when we do these addition experiments. So, and we do see functional effects with those. So, well, we think at least some of those are stable enough to make it through the pathway and end up in the appropriate place in the cell. But how many we lose on the way, yeah, very difficult to answer. We don't have an answer for that at the moment.
All right, so we've had some discussion of cell cycle, and I think that Marijke Zonneveld has a different interpretation there. Marijke, would you like to bring up your point?
Well, I was just wondering that I read that at least the co-culture experiments were incubated for, I think, 10 days with renewed donor cells, making the EVs, which would have the guide RNA. So, and even then you have lower, relatively low efficiencies of the GFP. So yeah, I would say that cell cycle is not then the most logical parameter that would define the GFP switch, I would guess.
Peter, is it okay if I chip in on this?
Of course.
Great. So I do think that the cell cycle idea in of itself is very interesting, and I completely agree with Marijke. In rare co-cultures, we have a co-culture for at least 5 days, so it'd be very difficult to check that. We could theoretically, of course, with siRNA affect specific targets to be involved in the cell cycle, but I think at that moment we need to be really careful because it is known for Cas9 activity that in different stages of the cell cycle, there's a difference balance between non-homologous end joining and homology-directed repair, and as such also a difference in the chance of getting mutations due to double-stranded breaks. So if we were to go down that path, not only I think would it be very challenging because of the time frame in which we do these experiments, if we were able to like maybe increase the efficiency of loading, for instance, for EVs like Peter said, then maybe we could synchronize the cells and perhaps introduce these EVs at different stages, but then we would have to, I think, alter our readout or our reporter assay, perhaps think about more of a like Cas9-mediated transcriptional activator or such, because I think right now, if we really start to look at those pathways, it does become technically a little bit dangerous because you could, of course, also be affecting transcription or Cas9 activity.
Yeah, and then Esther also wants to say here that the EV uptake could be fast and not dependent on cell cycle, while the efficient delivery of RNA could be dependent on a specific cellular process. So that's a good point. And then Tom Dredonks, you have a question about loading as well.
Yeah, I was wondering, once the small guide RNA is taken up in the recipient cell, what is the affinity for the Cas9 protein? Is that very efficient, or could that be another rate limiting step in your assay?
Yeah, good point, Tom. I wouldn't have the answer for you. It could be a rate limiting step, of course. Cas9 has to find the guide RNA in the cytosol and bring it to the nucleus. Yeah, it could be a limiting factor. We don't have a clue on this at this point. Like I mentioned, for me, the loading efficiency seems by far the most plausible explanation for this, especially considering how efficient the transfer is as you compare it to synthetic systems. But whether such other factors also limit the activity, it could well be.
Other than that, I think it's a great paper, and it's really cool what you can do with it, looking at uptake and everything. So it was not a criticism.
Thanks, Tom.
Just out of interest, do you see Cas9 in the vesicles from your receptor cells due to Cas9 is transported?
No, we don't see it, but I also don't think it in donors. From the donor cells, you mean?
Yeah.
Yeah.
From,
So
from the receptor cells. From the receptor cells, at least sort of interest, like receptor cells can.
We don't isolate vesicles from the recipient cells, but I guess at this, in a different, we're trying to do something similar. I think if we don't do any tricks, we don't readily see it in the vesicles. Olivier, do you know the specifics here?
Yeah, so we don't observe, so if we overexpress Cas9, we don't observe it in vesicles on Western blot, and I think that's in part due to the fact that there's a nuclear localization signal on there. But I mean, of course, that's also the case for the Krelok system. So it is, of course, still very possible that you could perhaps have RNA transfer, although it is, Cas9 itself is substantially larger than Cre recombinase. So we haven't used the system that way, but we have, for a different project, we do not see kind of this random incorporation of Cas9 into vesicles if we just overexpress it. But maybe we would have to do experiments with such a sensitive reporter specifically for that readout to have a more decisive comment on that.
Yeah. Yeah, I would also argue that probably the other way is way more efficient, like guide RNA into reporter than Cas9 out of reporter into donor and being functional in this low levels. But yeah. I think what I was like thinking as a second step is just like if you can get not only the nuclear localization signal, but also the Cas9 more in the endocytotic. Donors, I mean more like, is there any localization signal that could be attached to Cas9 to load them more into the vesicles in the receptor cell, so that then, or in the endocytotic pathway, so that then they see the guide RNA right when it comes in, kind of.
Yeah, but it still needs to make its way to the nucleus.
Yeah.
So the only thing I can kind of say about that is that we did see that for some cell lines, while we were generating these donor cells, that if you have poor Cas9 expression, the efficiency of the system goes down. So we do have high expression of Cas9, and that works. And we assume that indeed, the more Cas9 you have in the cell, the bigger the chance that the guide RNA and Cas9 find each other before Cas9 goes into the nucleus. So yeah, it seems to be important that they are really given the opportunity to find each other at that stage.
So I apologize, I cut out there for a minute. So I've lost my chat window, but I know that Zubair Ahmed had a question about labeling, another labeling question. I'm not sure, Zubair, do you want to bring that up?
Hi, thanks for the talk, that was very good. I just wanted to ask first, before the question, what assessment did you do to quantify the RNA per EV?
Sorry, I didn't completely get the question. Can you speak up a little bit?
Can you hear me?
Yeah, now I can hear you.
That's better.
Yeah.
Yes.
Great. I'm just wondering if you can label the RNA using any isotopes that can be detected by mass spec.
Ooh, I don't know, to be honest. I mean, the way we now use a system is that we let the cell express the guide RNA. So I think that is rather difficult then to do that specifically.
Well, just along the lines of the detection ability of the loading of RNA into every EV. If we do have an isotope or a marker for the RNA, we might be able to use correlative mass spec to look at the heterogeneity and loading of EVs. I don't know if that is of great advantage here, but it was just another way of looking at it.
Yeah, it's an interesting point. I'm actually not aware of it. Can you do mass spectrometry already on single EVs?
Yeah, that's kind of what we're doing now. We haven't really published anything. We're trying to do single EV analysis using correlative electron and mass spec that can go, that can give like a high chemical resolution and a electron resolution. So you'll be able to get complete information. But I don't know if you'll be able to identify your guide RNA specifically in each of those EVs. But if they're labeled, for example, just for assessment, just for the sake of assessment, you might be able to see the efficiency clearly at single EV level. But I don't know how much of use it would be at this stage.
That's an interesting thought. Well, maybe we should discuss that outside this.
Yeah, definitely.
This meeting, but I think it's an interesting approach. Yeah, like I said, the way we process it now is that we let the donor cell express the guide RNA, which makes it very difficult to label it. And it's also the loading efficiency, I think, is a bit low now. But yeah, I like the idea, definitely.
So I think with that, we will bring this session to a close. And I think we've come to some conclusions here. And one of our big conclusions, I think, is one that we always have, and that is that we're bumping up against the limits of sensitivity. So whether we're going to try to do some single EV mass spectrometry or try to look at subpopulations of EVs and their transfer, we have, I think we still have some work cut out for us to become a little bit more sensitive, or to make enough material so that we can look at different possibilities. But in any case, I think that we've learned a lot today about this new system and what it can do, what it can tell us about EV uptake. So I want to thank the authors, Olivier and Peter, for joining us. And most of all, I want to thank you, Franziska, for your very clear presentation of this paper and for starting us out with some great questions for discussion. And thanks to everybody else who was able to join today.